there. How are you? Okay, so let's start with arthropods, and this can actually be video 0. So I've been putting some of these up, and some of them have quite naturally been getting copyright claimed because I'm linking to these other videos and actually showing some of those videos in mine. So I can't do that anymore. They're going to take away all my coursework that I've done so far. So let's not do that. So that means you're going to have to watch the videos all by yourself on YouTube. Oh my gosh. Or occasionally on Vimeo. So let me just go through the different parts and show you how I'll show you how these things relate and where to watch them in each little section. I think for now we don't actually need to see any of these. We can go right into section 1, but they're all going to be linked from the arthropods, and I'll put my YouTube videos of the PowerPoints up here at the top, so it'll be good. Okay, anyway, let's go to this and make it big, which will turn on the subtitles. Yes, that's what we need. Okay, so arthropods. The name means jointed foot or jointed leg, I think is more accurate in this case. And these are the things that are the most common large animals on the face of the earth. There are more species of arthropods than anything else. These would include the insects. They would include things like centipedes and crabs and all kinds of other arthropods. Ticks, mites, spiders, and a bunch of really weird things that we're going to talk about in the lecture and you're going to see in the videos. They're so cool. You got to make sure you watch all of those. So what makes an arthropod? So that's what we're going to talk about in this first little segment. So this is a view through the arthropod shell. So it's actually going to be made down here. You're going to have an epidermis and an uncalcified layer. So the uncalcified layer actually starts out as mostly chitin. And the chitin is going to be the same stuff that the mollusks laid down to start out making their shell. And then on the outside, we have a calcified layer. So this is chitin, and we're going to rapidly recruit some calcium carbonate to this layer to essentially make a thin version of a mollusk shell around the arthropod. So they're actually pretty close to each other in the way that they build their protection. Then on the outside, you'll have a thing called the pigmented layer. And so if you have an arthropod that is blue or red or whatever, then that pigment's going to show up here. When you cook a crawfish or a lobster, it is going to burst some of these little pigment pouches in the pigmented layer. And so that's why the color is going to change. So you're just physically just denaturing this area. Then you got an epicuticle on the outside, sort of a nice shiny little protective layer. And as the arthropod grows, what it's going to do is it's going to shed the calcified hard layer and the pigmented layer and the epicuticle. And you're going to start off with just an uncalcified layer. So right after shedding that exoskeleton, arthropods are going to be very soft and squishy and vulnerable. Poor little arthropods. Yeah. But this is essentially how it works. And I think we've got crustaceans on the left and I think insects on the right, but it's the same idea. In each case, the sort of uncalcified layer or cuticle is going to be made of mostly nice little sheets of chitin. And we're going to mineralize with different minerals that cuticle on the outside. So if you live under the water, calcium carbonate is easy to come by. That's why so many things that live underwater make their skeletons out of it. On land, you'll have to store up other kinds of minerals in other ways, but it can be done. It can definitely be done. Okay, so for this one, what I'd like you to do is if you got the time and inclination, go out to the store, go to the freezer section, pick up a box of delicious crab legs, or go to wherever the heck you go and pick up some crawfish. Nice little crawfish dinner, and yeah, they're educational and delicious. I think that's the key point. And then once you get one of those, I can wait. Yeah, you can pause this. Once you get one of those, you're going to crack open some of the claws or some of the appendages, like the legs. And inside that, you will see the meat. So the meat that we eat from these animals is going to be mostly the muscle. Now, something like a crawfish, we also eat a lot of other parts, depending on whether you like to suck on the head or not. But yeah, the really good stuff is it's all just nice little muscle fibers. And so you're going to have extensors and you're going to have sort of contracting muscles, they call it flexor. And each of those little jointed appendages is going to be set up like this. You'll have these little cylinders on the outside. That's the hard shell. That's the exoskeleton. And then on the inside, you'll have your muscles and joining it up. So there'll be one side that's really close to the side. So this one's going to bend this way, and you can see it. That's the only way it can bend because it would run into the wall if it tried to bend outward like this. And there'll be this little rubbery membrane on there. It's kind of bendy. And as you're flexing the claw on your crab or whatever the heck you've got, you can see that part move, and you can feel the hard parts there. Those are the parts with that big, thick exoskeleton. So, yeah, delicious and educational. Wonderful. All right, so what about these muscles? So let's talk about muscles a little more. And so we're talking about the motor neurons. So motor neurons are going to be the ones that connect up to the muscles and actually makes them flex or contract. Flex or relax, rather. So in Cnidaria, they're going to use glutamate as the #1 neurotransmitter to make their muscles flex. But look down here. So Anthozoa, these are going to be like the corals. And do you remember the corals that we watched, the videos? So they're circular muscle, these big rings of muscle around the outside, can contract, but they have a contraction time of between 60 and 180 seconds. So yeah, you say boo, and you gotta wait 3 minutes for the Cnidarian to jump in surprise. Oh no. Yeah, that's how they are. Very, very slow. But their sphincter muscle, which is their mouth, basically, that beautiful colored thing in the center, that has a much quicker contraction time, so it can pinch down and close in on a fish or whatever it is that has wandered in or been harpooned and dragged in. So they're going to use glutamate to make those muscles contract. And they also have interesting chemicals such as beta-alanine and taurine. And all of those serve as neurotransmitters in Cnidaria. And those will all be things that'll make those muscles contract, we think. There are some things that aren't fully understood about Cnidarian muscles. Anyway, GABA, which stands for gamma-aminobutyric acid, is going to be one of the things, at least, that makes their muscles relax. And Cnidaria are complex. They also use some little proteins as neurotransmitters, which is something that we do in humans as well. We have a lot of these neuropeptides. And if you had my biology 2, we would have talked about some of those. Anyway, mollusks use acetylcholine for their neurotransmitter on motor neurons, just like we do. How cool is that? So as is the case like chordates like us. And so here's some bivalves and gastropods, and you can see they've got a little bit faster contraction time, 1 to 2 seconds. So, you know, they're going to be a little slow on the uptake if you tell them a joke, but they'll laugh eventually. You won't have to wait 3 minutes. Arthropods are going to use mainly glutamate once again, so they're going to be on the simple side. And you will remember that ctenophores were glutamate only. They didn't seem to have anything else going on there, just glutamate and aspartate. And so Cnidaria and arthropods are still keeping it sort of simple, and they're using that. Mollusks and everything up to chordates do have glutamate as a neurotransmitter. It's just that we have all these other ones that tend to be used for more things. Once again, GABA is going to be the inhibitory neurotransmitter. So that's going to tell the muscle when to stop contracting and relax. But look at these arthropod muscle flex times. Look at that. 0.1 seconds, 0.025 seconds. Yeah, that's really good. That's on par with human sprinters. Maybe a little faster. So, yeah, an insect flight muscle has to go really fast twitch time. And it does. It absolutely does. Okay, but what's interesting about arthropod neurons is the way they are built. So shown here, let's see, invertebrates going to be at the tops. These are going to be our different arthropod neurons, are going to be like A2 and A3, if I remember correctly. They'll have some A1s. People like us, vertebrates, will have neurons that look like this. So I want you to look where the yellow thing is. So the yellow thing is called the soma. This is the body of the neuron, the chunk that has the nucleus in it. And then at one end, you're going to have neurotransmitters, and at the other end, you're going to have little dendrites that can pick up different signals. So we got signal comes in, signal goes out, just like that. Yeah, pretty nice. But in human neurons, you'll see that most of them have the body of the cell kind of integrated right in there in the middle. And so all the little dendrites are going to come out from the actual cell body. But look in arthropods, the cell body is out here at the side. And so they will have things that are receiving electrical signals and transmitting electrical signals on the rest of the cell, but they take that nucleus out of the loop because the nucleus isn't actually going to be involved in electrical transmission. If anything, it might even slow down that transmission. And so, yeah, there's a little shorter, a little more efficient circuit there for arthropod neurons. But what this means is that you'll have bundles of fibers, like nerve fibers, and those will contain this part, long and stringy. And then you'll have these bunches of the cell bodies, the nuclei essentially of all the neurons, and they're going to be bunched together in these things called ganglia. And we're going to look at ganglia on the next slide of arthropods. So here, if you were to dissect away certain arthropods and leave only the nervous system, this is what you would get. So B is for brain, right up here at the top. And so you can see we've got a nice little bilaterally symmetric brain up here in all of these. But if you look, everywhere that legs come off, there's going to be a ganglion and multiple ganglia all the way down to the bottom. So from this, you can kind of get a picture of what these are. So let's look at this. So what do you think we got here? What would have this many body segments? And usually you're either going to have mouth parts or legs coming off of each of these ganglia. And so the answer is, of course, something like a centipede. So they're in the group called Myriapoda. And yeah, that looks like a centipede if you take away all the legs. So attached to each one of these little neural branches are going to be the leg muscles. B is going to be an insect. You're going to have these up at the front that are going to operate the mouth parts. And then you're going to have these, in the middle are going to operate the 6 legs. So we'll have 3 little bundles like that. And then all down in the abdomen, you're going to have another set of little bundles because a lot of them have abdomens they can flex independently. And the brain is here, and that's going to be hooked up to the eyes right over there. And this is a different kind of insect that just has those 3 ganglia, 1, 2, 3. Let's see, crustaceans are going to be over here, so something like a crab probably right there. And more of a long, skinny crustacean with more legs. This might be more like a lobster. They would normally have 10 legs. And so, yep, looks like 5 branches on either side up here, plus those little swimmerettes, like the little tiny legs that you get down toward the back. And then let's see, Chelicerata. These are going to be things like spiders. And we've got different kinds of neural architecture. But, well, at least 4 of these ganglia on each side are going to control the legs, presumably. And the rest are probably controlling the chelicera, which are like a modified version of a leg that's up by the mouth parts. And they have some really interesting little palps and things, little mouth parts on the front. And so that's how they do it. And that's a spider brain versus other arthropod brains. So the thing that distinguishes them is that they always have this big long loop. So it's like a big long jump rope stretched out all the way top to bottom. And all along this, they're going to have these ganglia. And everywhere that you have ganglia, which are all those nerve cell bodies, you're going to have something like legs coming off. And that's how they work. And so if you wanted to take apart these ganglia, so you could look in insect ganglia or a crustacean, something like a crab ganglia. And if you were able to section these things open, this is like a crossways section, this is a long ways section, they actually each have these separate little zones in here. And so each of the zones have names, and so there are different kinds of nerve cell bodies in each one of these ganglia that go out in different places, but they're always put together in the same sort of way. So there's a particular organization for the ganglia in arthropods. And you get slight differences depending on which kind of arthropod we're talking about. You see the insect is not exactly the same as the crustacean, but you got a lot of the same colors in a lot of the same places. And generally, yeah, it's pretty similar. And you can tell these guys are related. Okay, so that's the end of that one. And yeah, please come back for the second part.